and it can loop back and help us. Now, come to the title of this talk. I talked about 2 papers from 2018, a sort of pivotal time in my lab. I was just promoted to with tenure, and it's going along. I'm really happy. But our main procedure looks something like this. We have an idea of what we want to find. We're going to come up with a model that finds those. We'll do some sort of statistics and maximum likelihood to refine them. We'll detect some sort of points. On those points, we'll have another maximum likelihood statistical kind of operator, and then build them up to a higher order object and reason on them, and end up with the spinning brain I just showed you. But at the same time, the idea of how do you take this network architecture that's totally data-driven and allow the data-driven ideas to work on targets that were really previously inaccessible, very squishy abdominal structures as you move your belly around, as you eat, as you defecate, all of this kind of stuff really deforms what's going on inside, much more than the brain in a bucket. It started to work, and we were able to train on models with much less data and much less sort of fundamental numerical understanding of the processes. So this is really exciting, but it's not diffusion. I'm getting there. Now, why do we connect with diffusion? Well, there's a lot going on in Alzheimer's disease right now, and this has been part of my life since about 2004, my research life, getting into the Baltimore Longitudinal Study on Aging, which is at Hopkins, as we mentioned, or next to Hopkins. It's in Harborview at the NIA, but deep collaborations of understanding what's going on in all of these people. Well, about 6.2 million Americans 65 and older have Alzheimer's disease. That's 1 in 9 people. It's a tremendous public health impact, and it's a tremendous sort of problem with aging, with a lot of non-successful clinical trials. So how do we move from all of these trials and all of these data to something that can help us push forward? Well, it turns out that white matter is deeply involved with what's going on in aging. It's highly associated with APOE status, which is one of the big genetic risk factors. And it is predictive of sort of both longitudinal changes and future conditions with vascular burden and APOE status. If we start mapping what's happening to people over time, people who not are impaired now, not are impaired in this study, but in the future who get impaired, have changes in the white matter microstructure and have changes that are not volumetrically visible yet. So this whole idea is that the gray matter sort of computers that are breaking down or the wires that are connecting them, that hypothesis was really oversimplified. We need to drill into the interactions of the brain as a system instead of just saying, oh, it's a chip and a wire. It's a lot more complicated than that. And we need the tools of diffusion imaging to go after why. We're starting to map out what are these tools? What are these bundles? How do these things change and age? And if you plot these across a lot of people with the tools that we currently have, we find trends and shape changes both in the microarchitecture, sort of FA is a canonical representation, as well as sort of the ending radius and the termination, which is interacting with volume and length change and the fibers themselves. And all of these things are just starting to become possible. So we're finding these associations. They're highly predictive of what's going on. And I'm showing 5 people, 5 scans of the same person from 74 to 82 across there. That's a lot of changes happening. It's probably fairly unlikely that the tract doubled in volume over this time. So how do we get tools from this group, this community, our group? It's, I'll take responsibility. I have some of our tools there too. How do we get these to be more reproducible, more robust, so that we can tackle these really big public health challenges? And for big public health challenges, I'm really mentioning our large-scale national projects that are multi-site, multi-center, multi-generational in terms of the scientists who are running them, integrating genetics, molecular biomarkers, structural biomarkers. Here, look, we're up there. We're just a footnote in the 3rd panel over. I want to make sure we're not first and last. We're way up in the middle. And cognition and thought. So to do this, we're building consortiums with NIAGADS and the ADSP to bring together the sequencing, the functional harmonizations, and the AI/ML deep learning, all of that learning data-driven stuff. Now, we're not going to solve all of these problems. I'm not smart enough to do all of this myself. It's going to take a community effort. And for the community effort, we need ideas to bring together and have the experts in each individual sort of subdomain curate the data and release them to everybody. And then through this national scale effort, the experts in machine learning can team up with the experts in each of our sub areas in terms of fluid biomarkers, cognitive. I don't need to read everybody up here, but we have a large team of sites that are each harmonizing a tremendous amount of data and bringing them together. We have a target of, by phase 3, 70,000 individual people with a tremendous number of biomarkers across all of them, and making all of these harmonized and compatible. Again, I want to emphasize, we in the diffusion community are 3/4 of the way down as an important marker, as an important piece, but a piece of this overall puzzle. So we need to figure out how we can make, quote, normalized images, the markers that we need in specific tracks, as well as free water adjusted markers in all of these tracks. What does that mean? How do we get there? And how do we let then the machine learning people go across all of these different markers? So, phew, I got to diffusion. It's 7 minutes in, but this is why I'm doing this. This is why I'm involved. Let's take a step back and think of diffusion as a sensing process. And again, Kurt Schilling is on the call. I stole this from the next couple slides from him, adapted, cited, whatever. We want to take the random motions of water and prefer either a virtual histology, the microarchitecture, or this macrostructural in vivo brain dissection. And to do that, we're going to visit Nashville here, and we're going to drop in a little bit of hypothetical water, either in the library, in the downtown party area, or out in the river. And when we see what happens is when you drop something in, either you're tracked in the stacks in the library and it sort of stays within the building, or it spreads out in a random walk through downtown, or in the river, it sort of bounces all around, because different processes are governing what happens in a library, what happens out on a river. We don't jet ski, it's not that pretty, or what happens in downtown. And by looking at those patterns of motion, we can fit the types of structure. Now, as we say that each sensing is observing only one direction, we get an image, we have a signal, it comes along one of these directions. We could have a different direction, different angle, and we have an attenuation model. Sorry, my slides are not advancing. Advanced slide, there we go. An attenuation model that says, here's a little bit of math. This is the apparent diffusion coefficient in a particular direction. Let's take all of these measurements and fit some sort of model. But it's really important to realize that a diffusion experiment is not just a one thing done. It's not just this one. I could have run the one I just showed, or I could have sensitized a lot more, or a lot more, and I would get very different images. These are different projections, different snapshots of the anatomy that are highlighting different types of structures. It's not just brighter and darker. We're getting different types of information. Now, as we want to take this information either from a B value, from the directions that we're sensing in, we can fit a model. And of course, a tensor is the right answer. At least this was very exciting when I was doing my PhD. But a lot more has happened in terms of constrained spherical deconvolution, cue ball, diffusion-oriented transforms, angular structure, shard, multi-tissue CSD, forecast naughty, but lots and lots of different models. We can compare these models and see that different types of structures on the same sort of data yield different qualitative interpretations of the tissue. Whew, this becomes hard. And I spent a lot of the last 5 years implementing, comparing, and evaluating these models. Now, if you'd like to implement these models, it's a lot easier in Dipy, and we have a lot of talks now on how you can take these, explore what the different models say about your data. And then remember, just as George Bach said, all models are wrong, but some are useful. So which of these models are useful for your population and your study set? When we have those, we can build up models, look at what's happening in an intravoxel structure. So as long as we are tracking, create a set of fibers that represent the brain, and then try and tease apart what's going on. Okay, so that's why we're going after these. These are sensing processes, these are estimation processes, and we have this grand vision of trying to get this on 70,000 people. How do we get this harmonized and appropriate across everybody? Okay, here's the brain. Our lab is looking at anatomy and ontology. Are we looking at the right tracks? Do we agree on the names? Do we agree on the structures? Can I compare things? Because if we just use text instead of some sort of agreed upon prototype, we don't know what's going on. Second, as I mentioned, these imaging acquisitions are different physics experiments. We need to think carefully about when a physics experiment is sensing different types of underlying structures. And the answers, the same metrics from those models, are going to mean different things if different physics experiments were played out. And then all of these have approximation after approximation after approximation. How do we know when those approximations are appropriate? How do we know when they're applicable? And how do we get them into a form that we can then pass off to the machine learning experts who are going to do multimodal, multi-view types of inference? So bringing these all together, our lab is focusing on different models of reconstruction, variability in definitions, and consensus on the white matter pathways. Kurt has a fantastic slide of all of the different things we could look at, all the way from acquisition all the way up to interpretation. And we are slowly picking at these in terms of where can we make a fundamental difference? Where can we address things that are at the height of a problem or not? And we could give a talk on any one of these boxes. But I wanted to highlight something that when I was preparing this keynote, went to my original job talk at Vanderbilt as a fresh postdoc saying, I'd like a job, please. Well, quantitative diffusion MRI is not new. We were formatting likelihood-based approaches with similar sort of how do we make them robust and how do we interpolate these things back in 2009. And it was cutting edge then, and we're still having these problems. But the big change is now we can do data-driven inference instead of only model-based inference. So as I said, we work in different parts. So I'm going to show a non-brain. This is a slice of a lung, and you can guess that little spot there is non-good. How do we take this slice of tissue, look at imaging features and clinical features, and get into prognosis throwing that in a GPU? Great. We see a lot of computer vision. We see what's happening in NeurIPS and CVPR and all of these places of beautiful models running on hardware. Well, if it were as simple as let's take a 2D picture where we can sample it to 224 by 224, throw it to some canonical network we grab on GitHub and get an answer, we'd be really happy and we'd be done, and I wouldn't be giving this talk and you wouldn't be listening. Rather, we have an entire stack of 3D imaging volumes of varying contrast with all sorts of artifacts. Those gradient volumes, the acquisitions, are different based on what was intended to play out in the scanner's coordinate system versus what actually got played out in the scanner's coordinate system versus how I moved my head while I was in the scanner getting scanned with what actually was being played out versus what was intended to be played out. So it's a little bit of a hot mess to try and make something that's reproducible across all of the types of variations that happen in these protocols. And it's even worse than that, because when you start looking across all of these studies, the types of acquisitions, the actual b-value, if it's a single b-value, or if it's a multi-b-value experiment, which b-values were played out changes, as well as the types of shells. Phew, we haven't solved the problem fully yet, but I'm going to spend the next about 30 minutes going through areas where we've made improvements and we are making tools available and where we hope to go. And the first is in distortion correction. When we run fast images, we get sloppy spatial consistency. This is due to the EPI readouts and local field homogeneities. These appear as little divots in the brain, and they affect the fiber trajectories throughout the brain. We might think, oh, there's a little divot in the prefrontal lobe. When I was in graduate school and training, it's like, we just don't track near the prefrontal cortex. It's probably a 3, 4, or 5 millimeter divot. Give it a centimeter. We don't track up here. We're done. Unfortunately, now that we've gone and asked, do those change? The answer is actually, yes, there's bundle changes throughout the types of processing if you had a properly geometrically constructed thing versus not. At first level, it's about the same. It's mainly in this area, but there are subtle shifts and alterations of microstructure, of volumetry, of ending points, of all of these things that happen that are propagated throughout areas well away from the local field homogeneities. So our aim is to be able to use deep learning to go back and look at the legacy data sets, apply what's going on, and as I said, with deep learning. So, okay, how do we do this? Well, if we run a single scan with a bandwidth of about 1 kilohertz, you get something like this, which again has sort of the prefrontal divots and areas of distortion near the acoustic penises. Now, if you run the opposite way, the fantastic team at FMRIB has the fMRI software library, and they have top up, and so does Karopir with distortion correction in Tortoise, Dr. Buddy, and a number of people have figured out that if you run these in different ways, either right, left, AP, PA, you can say, these distortions either opposite or contradictory or relate to some underlying hidden object that you can find. We can apply top up and we win. Okay, but our fundamental problem is if people were acquiring all these data, we would be happy little researchers and move on and not be talking again. The problem is we got a tremendous amount of legacy data or data where one of them has failed and they tried to run an APA and they got an APP twice or something like that. So what do? Well, what if we took this image, took the MP-RAGE or structural image that we have, and instead of just doing image registration and trying to match up the pixels because they're not exactly the same material, there's different things that get shown, what if we tried to synthesize an infinite bandwidth image? And we label this synthetic distortion-free B0 as a infinity bandwidth. Now, it's not a perfect image, right? Image synthesis is problematic. I can have PSNRs that are high or structural similarity indices that are great. I'd be really cautious about using this as data itself and trying to fit a model, because that's going to be really driven from the T1. But if I use that infinite bandwidth and then only correct the left one and throw away the infinite bandwidth one, I can get something that looks incredibly similar to as if I had the actual true reverse phase encode using the model-based approach of top up. So in this approach where we fake the infinite bandwidth, we call this symbodisco. Now, okay, how do we fake things? Well, our key idea was using image synthesis, and here's the conditional adversarial nets, where you take a map and you generate an image of a city or vice versa. If you want to switch day to night, if you want to put handles to bags, I need to put the one on here where you can make everything a cat. Sorry, I'll update my slides for next time, but you can make everything a cat. You want to have square cats and round cats, anything you would like, deep learning can make it look like it. Our idea was, let's take an input. We'll try to make it a B0. We have a lot of true pairs. Let's see what happens. And we put this in terms of a private data set, the Human Connectome project and the BLSA, 3 very different acquisitions from about a 2 millimeter to a 1.25 millimeter to this highly anisotropic. The true image in plane resolution was 2.2 millimeters isotropic, on the scanner upside to 0.83. So very different acquisitions and very different data sets that are available. Taking these, putting them in a pipeline where we take the distorted B0 and the T1, synthesize an infinite bandwidth image, apply top up, which is just the regular old tool from the fantastic folks at FMRIB, and get a corrected image. When we do that, we find that it works in terms of mutual information just as well as top up and synthesis. In terms of PE, we correctly match the processing when we had the reverse phase encode available, and we find highly reproducible images on scan rescan data sets. Okay, that's great. It's nice. We tried it on our 3 training data sets and it works. That's sort of typical. But then we went and grabbed a whole bunch of data sets off the internet that are available and started looking at, does it work across highly different variations in T1 and B0? And the answer is yes, it works really stably in that we gave it enough training data that span a very wide range of clinically feasible data sets. And so this tool is available now, we're happy with it, and we're working with the folks in Dipy to get it in there. So, okay, distortion correction is sort of a spatial thing that reminded us of segmentation. It's a little bit different, but what about the actual models, right? Can we get in there? Well, our first insight is there is actually truth. We can go and we can do histology, either 2D histology or 3D histology, and we can say, this is what should be there. There is a data-driven answer. Fortunately, one of the experts here is here at Vanderbilt, Adam Anderson and Ure and Kurt again. And you can take a in vivo image, pair it with an ex vivo image, pair it with a block face photograph, which then has light microscopy, which then has confocal microscopy, which then in this 3D image, you can construct it. And we can say, this is a histological FOD that should be there based on whatever analysis you would like to do with that tissue structure. And then we can say, how does constrained spherical deconvolution or some other deep learning models align with the true? And our data-driven ones should be better because we can do it. Great, this makes sense. Let's go ahead. The problem is, how do we do it? Well, with 5 years, we got 567 voxels from 3 monkeys. Okay, these are incredibly expensive. We can go ahead and train a model and do data augmentation, all the computer science, low shot learning type approaches, and we can do okay. But that's still only 567 data points, a bunch of them that we're going to need for external validation and training and internal validation. So we're going to be training down on 400 single voxels. And that's going to be really hard. So what our idea was, can we use something else? And Vish came into this project and said, look, these blue ex vivo data sets are incredibly expensive, but we've got tons of scan, rescan data laying around the lab. I was running a project at the time, having traveling subjects going around all these sites, scan, get in the scanner, get out of the scanner, do all of those kind of things. We've got lots of those. And aligning up myself to myself 5 minutes later is non-trivial, but it's totally doable. Let's take those pairs and put them through the same network at the same time as the truth. And if we do that, we have the general loss function, which is just, are we the same as histology? And we got this other channel down here, this blue and green, where I don't know what the answer of me to me is, but I know that it's the same person in the same at the same time. And I can add this secondary loss function, which is a contrastive loss. At the time, we called it null space tuning. This was like tuning a linear null space, but we lost the naming game, and it's actually now called contrastive loss functions. So in this contrastive loss function, meaning only the difference, not relative to the true, how well does it work? Well, if we blow up this on here, we have a sort of consistent level approach. And if we blow up the other one on the other side, over here, animation, go, there we go, one. This is with the deep learning. This is with constrained spherical deconvolution. Goes down here. We get highly similar answers. And we can see that the same kind of things are happening. But maybe it's not as robust. Maybe it has some problems. Let's take this network, the same idea of a contrastive loss. And in addition to just saying scan, rescan, what if I take 2 different sites? Now I have the same person at site A and site B, and I can still throw these things in the network. But now my contrastive loss function, instead of being Bennett to Bennett, is Bennett on site 1 to Bennett on site 2. And if we want to look at how that happens in terms of ACC, we have across sites and not across sites, massive improvements in the angular correlation coefficient, so the angular agreement between these FODs. CSD is reasonable, just regular old deep learning is slightly more consistent. And if we go to the null space deep learning work that puts both of these together, it dramatically increases the consistency of the scan rescan pairs across sites. And if we look at this again, the color behind the glyphs is the scan rescan reproducibility of constrained spherical deconvolution, a lot of darker voxels in there. At DNN, it's a little bit better, but has some pock marks. And at the null space contrastive loss function, it goes up quite a bit. And we can really focus on sort of these areas here on the boundaries of the main white matter tract where the non-tuned methods have more problems. Okay, that's nice, we're getting there. We have these types of approaches. Can we change the way we're learning so that we can go across p-values? Well, rather than learning on spherical harmonics or something like that, we then need to start learning on Shor basis functions that include the b-value. And so if we just use regular spherical harmonics, we get reasonably. If we use Shor without optimizing its basis set, it works pretty well across p-values. And then if we optimize it, we get this green consistent relationship. So we can actually learn the same sort of angular structure across different sets of B values and allow it to be trained with one, be 6,000 and tested on 3,000, 6,000, 9,000, et cetera, and allow the same estimator to work across different B values in a consistent manner. And if we look at what that looks like, again, as a glyph in the frontal white matter, things look reasonable. This is the kind of thing that we would expect, and we can tune these to be more consistent across sites. So does this work across other types of designs? Well, yes, if we take this residual network and we start applying it to tractography, we can get very reasonable ideas of what works. And the issue is when we start to look at multi-shell designs and multi-shell areas, we get to the problem of indeterminacy. And if it's a single indeterminate type of approach, I can't tell the difference between a higher diffusivity and a two compartment type of thing. But if I factor in patches and I start allowing patches of diffusion signal and ask for local spatial consistency as well as model consistency in my learning process, then I can get back up to this multi-tensor constrained spherical deconvolution, the multi-shell CSD type of approach, and allow more robust learning across single and multi-shell designs. We can also take that same contrastive loss function, start applying it across B values. If you ever looked at somebody who's been scanned, rescanned 1.5 versus 3T, the types of sequences available are very, very different. And getting something that is consistent is much easier with a contrastive loss function instead of trying to put in a regressor or a control function or something else. And this can make a big difference in terms of where we are going in harmonizing these models and in making things consistent. So Vish graduated and Colin came along and said, this is nice, but we're previously really only taking approaches that harmonize a single voxel or a single data set. How do we think about the whole acquisition type of experience? The distortions, the bias fields, the gradient nonlinearities, could we consider everything in an end-to-end framework? And the folks at Mushak, which is a Mackay challenge, came up with an excellent type of idea. They released a challenge of, on the connectome scanner, a state-of-the-art acquisition and a standard acquisition, and the same thing at 1.5 and 3T across, I believe, 5 subjects. And so very different acquisitions, very different, very high level, 50% error, scan, rescan types of fits on FA. Again, very high error in terms of this is a Baltimore longitudinal study of aging, 1.5, 3T, same person a year apart. Very large differences in the types of microstructural measures you would see. And when Colin approached this, we said, okay, we can do a supervised approach or an unsupervised approach, a semi-supervised approach, or a semi-supervised contrastive learning. And this is what Vish and I were really working on in terms of how do we have this other type of information? Well, we then started looking at multi-target and unsupervised and multi-target contrastive supervised. So how do we get into this bottom area where we have all of these scan, rescans, but we also want to fit all of these different types of models? And to do that, we started latching on to the Shor estimator. It's a spherical harmonic type of representation of the basis functions that additionally has a radial Q space component so that we fit the B values simultaneously. Dipy has this in there, and the default and parameters give you about 50 coefficients, and it's simple to use, gets us transforms. I highly recommend it in order to access these consistent representations across the subjects. And we rely on these implementations in order to take the experimental structure, compare a baseline 1.5 scanner with the 3 Tesla scanners, look at a state-of-the-art versus a standard sequence. This is the same scanner, different acquisitions, versus a 2 different scanner, a regular scanner of a state-of-the-art versus a standard acquisition, versus then the connectome versus a standard and a state-of-the-art versus state-of-the-art. It's a lot of pair ways we can look at this. So 4 subjects here, 5 subjects on VLSA. a clinical B value, and then a multi-B value type of thing. How do we get a single type of approach that is going to work on all of these very different data sets? So we started with a CycleGAN, which basically takes a generator that says, how do I take a learning input, one of these guys, and produce a target space, which is another scanner, and then create a second generator that goes the other way, be able to make a discriminator that tries to detect when an image is faked or not, and then have a generator that goes back the other way and detect whether that is faked or not with a different function. So it's a ping pong type of approach between faking one from the other and faking the other from the one, and trying to make the generators good enough to fool the discriminators. Okay, so we can do style transfer, and it's a great idea. Several people put this into the Mushak challenge, and they actually didn't do terribly great. And after that challenge, we sort of tried to figure out what was wrong with this core structure. Well, this core structure is basically estimating this one set of acquisition features, this one set of parameterized network. So Colin said, Willie, there's several things going on. We have both an acquisition feature space, and we have this sort of subject feature space in terms of the anatomical features of the fundamental geometry. If we broke apart the network to encode the anatomical features, encode the acquisition features, instead of just synthesizing it, but let's encode those and then generate an image and separate this encoding and generating through a specific set of latent feature spaces, and then having the discriminator model. This no longer is a GAN model, but it's rather a disentanglement model. Can I figure out where the sources of variation are by putting in these layers of physical knowledge of where the structures are, encoding knowledge of what types of parameter spaces make sense, parameter spaces for the encoding, and then a generator that can take those and produce images that are detectable. So these new parameters, these beta IJs, are what are new here. And if we look at this, and we take the target image of connectome and we want to come from a prisma, and CycleGAN Shore is just, hey, let's use Shore and we'll reproject. There's a lot of error. The corpus callosum is kind of okay. If we use a regular CycleGAN of, like, this is the first thing I tell the students, hey, CycleGANs are great. It can make cats into anything. We hit a wall, and it's just like, it kind of works okay, but ignoring all of this GAN framework is better. If we then take that complicated framework of disentanglement and we apply it on a slice basis, we get very highly consistent white matter. And this is using all of that co-spatial information from the slices to be able to drive that learning framework. Now, there's nothing in that model that said it had to be a slice. If we do with random patches and 3D patches instead, it does better than the Shore, but on this example, not quite as good as the slice. And we really look at that in the corpus callosum here, and the sort of posterior white matter, gray matter areas. Now, if we go the other way and say, let's try and use a connectome to a prisma, we get really terrible GAN type of results. But the Shore wasn't that bad, but the patch and the slice, these disentanglement models using the Shore basis functions, really are able to learn a lot more and get things much more consistent. And interestingly, going in this direction, the patch is superior than the slice, and we're really working on how we can get these representational functions to make sense and be most applicable across the wide variety of types of data that we all are acquiring. Okay, so I've talked about distortion correction. I've talked about harmonizing models. Deep learning is sort of applicable in both. Let's talk about connectomics. Well, we've got a set of words. They're in English or Latin, or I don't know what language. We slap English and Latin together and then translate them to other languages. But we've got a lot of different definitions. And so Kurt and I were arguing for a while. We made a protocol called Tractography. We got a lot of people together, and a lot of people argued with our paper that they didn't like this or didn't like that. And so Kurt led an effort and said, working with Francois, let's do a lot of people who are all experts in their field. Let's give them 6 people, we didn't tell them it was actually 3 and 3, that are a scan rescan pair. We'll do all of the tractography and just give you the fibers. You tell us if you know what a pathway is for up to 14 pathways, and find it. Now, only do the pathways you know. Don't tell us the ones you don't know, and it's fine if you don't. So we got a bunch of groups who are experts in their field who've published on their tracks to go ahead and look. Let's take a look at the results from the corpus callosum. From the internal capsule. This is cortical spinal tract up there. Looks great. What do you think happens? Here's one. Doesn't look bad. But if we go across all the groups, here is each little slide of this. Hopefully the video is working for you. Is a different group. Sitting here, chilling out. The tops are the actual fibers. The bottom is the maximum intensity projection fiber density. Anybody seeing beauty, consistency, everyone agreeing? No, it's a hot mess. So the problem is the interprotocol variability, the differences in what people define for the major cortical spinal tract are massively different. So let's go to something a little trickier. And so here's a superior longitudinal fasciculus. And we provided the same fibers. This is not differences in pre-processing. This is not differences in tractography. It's not differences in model fitting. It's here are the fibers. Go in and select by whatever method you like, which yours is. Let's see what happens. Oh, go back. I didn't play the video. I'm going to play the video, please. There we go. Actual results submitted by people who know what they're doing. A lot worse in terms of consistency. So we're seeing the interprotocol variation is much worse across the different tracks. And if we look at the protocols of deep water, even the core bundles, there is very limited volume-wise agreement. This is where 75% of people agreed that those are the core bundles. And on the streamline agreement, nobody picked the same fiber 75% of the time. And these were given to them. It's not like, oh, it's close. These are actually given from what we said. Okay, it's a little bit exasperating. We were hoping for better news. But given that we're here, can we use deep learning to build agreement, to establish definitions, to allow us to speak together more clearly? And Colin teamed up with Chi on this one, where we had 3 different data sets, recognizing the pattern, internal, HCP, and the BLSA, did track-specific bundle diagrams, and generated what is going on for all of these different protocols. Now, I'm sorry, there's not just one. There are a lot of different types of protocols. We picked 6, TracSeg, RICO bundles, Tracula, Extract, AFQ, and AFQ clip. We ran them on all the data. We made an atlas of where all of these intersect all of these different cortical surfaces. Phew. Now, that's a tremendous amount of data. So over 2,000 subjects that have been tracked and QA'd and aligned and okay. We could average this into an atlas, but then we'd have a really blurry atlas that people would argue about. Could we take this and make it a personalized basis set? Well, we had some success previously in the brain doing these tile-based approaches where we would, break the brain up, figure out what volumetric label went in all the places and make those spinning brains. Let's take that same approach, but instead of trying to figure out what the volumetric label is, let's put in which white matter bundle it was. And let's do that for all of the different bundles that are overlapping and see how well it does. So if you do multi-atlas segmentation, which tends to be my favorite, the best we could do was about 0.7 dice on all these big white matter bundles, with down to about 0.4 for the AFQ type of methods. Not great in terms of its accuracy. If we used a single atlas, it was surprisingly more stable depending on where we thresholded it, but still not great. But using this deep learning, we could get a fairly accurate reconstruction of where these labels are to provide a quick lookup of, do you agree that this is this label? And then we can do this with a highly consistent manner across subjects of, this is where these groups would call this particular structure from TracSeg, RICO bundles, Tracula, and AFQ, not necessarily using, well, not using the white matter, only from the T1. And we can use this to allow folks to have a communication about which definitions are appropriate. And if they don't like their definition, then adding in a definition that can then be used to say, this is the type of structure that we are talking about. So we've shown that we have deep learning in each of these places. We are working with data-driven estimators extensively and looking at all of the different possibilities. We're excited to work with other people who are interested in data-driven estimators. And we're particularly excited to have, now I did this last night, so now it's 13 days ago. We had the pull request 2520 having a residual deep neural network inference in. So if you'd like to use a neural network-based approach, it is in Dipy. And there's code in there that you can plug in your own data-driven approaches that we can compare. And we have a bunch more harmonized approaches that we are looking to get into Dipy for consideration and in for discussion of how should these be used? Where are their limits of consideration? Where are the areas where these can be most impactful? We've got a lot of other tools. We're working on getting everything else I showed earlier in today's talk into Dipy, into a way that people can use and manipulate. We've been working on harmonizing diffusion, validating tractography, looking at microstructure, lots of quality control and pre-processing, which is interesting to get into Dipy because getting the Python libraries consistent across all of these tools that everyone has been using is a bit of an art. I appreciate everyone's insight on this call, especially Principals and Ellis. Looking at getting cortical consistency along with everything else. There's a bunch of interesting statistical approaches where we can bound bias and diffusion. Looking at, again, the reproducibility of tractography and coming back to the fundamental signal of what is the right way to think about this as a physics experiment, even if we're doing a data-driven physics experiment. I'd like to thank the tremendous number of people who've made this possible. Our lab has grown outside and coming in here. I'm looking forward to taking a together picture outside again in a week or two. Tremendous amount of support by the NIH, NSF, and others. And I'd like to open it up for a little bit of discussion, especially on what are we doing to calibrate? How do we improve microstructure estimation? What does it mean to have data-driven techniques? And I'll end with questions, hopefully on time here.
Thank you. Great talk. Great talk. Thank you so much. Can you please start asking questions to Dr. Landman?
I can't see the chat easily on my screens, so if people only want to put it in chat, if you could read it, it'd be great.
So, okay, so no questions.
My email is
I think you
somewhere.
have done so much, Bennett, that like people realize, okay, so much. They have to process this information. So I can ask a question that I found really interesting. All three parts of your harmonization work, and if you can go back to that figure where you talked about the third method, and I would like to ask a bit about this discriminator. If you can go at that region, yes, consistent connectomics, and then that disentanglement model. Can you explain a bit more what is going on in this figure? So I see you have the labeled scans and the paired scans. So those are same subject, pairs of same subjects.
Right. So we have a architecture of what is going on for each scan, right? And we're working in the imaging domain instead of the estimator domain. And so how could we adapt the imaging to say, what is going on? What would have this person looked like in a different scanner? And so this is a method based on the method out of Hopkins for harmonizing T1 and T2 contrasts, because we have the same people, but we have different, we can go across people and we can go across imaging acquisitions.
Okay, great. Another question I have about...
Excuse me, may I have a question regarding this? I am Paolo Avesani.
Please go ahead.
Thanks for the talk, really inspiring. I would like to understand overall in this investigation on subject variance, what is the role of registration of the different scan?
So when we use rigid body registration to register between subjects after we do all the distortion correction, so within a subject. And then we align those to affine space in MNI to keep approximate, to reduce the variability, because otherwise we'd have to do a tremendous amount of data augmentation for all the different poses that could be happening. And then once these are learned, we take them back into subject space by reversing that affine transformation.
Okay, but the question was, okay, what do you believe is the interaction between the generalization you try to do over the subject and the transformation, the distortion of the brain because you bring in the same, necessarily in the same space? So from a certain point of view, you, let's say, you introduce an alteration of the brain because the registration, and the same way you try to generalize, to summarize the difference, but these differences are partially coming from the registration process. So just to understand how do you believe these two kind of process interact together?
I think it's really important to know when and where you are conditioning your network and when and where you're conditioning your modeling. So is the model conditioned on being in a similar space or not? Or is it conditioned on something arbitrary? So if you don't do any alignment and then you run your registration, run your data-driven learning process, you are implicitly using the alignment of the scanner tech. And you're implicitly saying that everybody is like this. And then if anybody is off, any angular structure or angular variation isn't going to be caught, and it's going to make your method far less robust. So either you need to make sure that sort of all possible axial types of scans are included, which is, you know, lots of rotation of the brain versus a moderate amount of right-left pitch of the brain in the estimation process, and that's fine, you could do that. Or you need to say, I'm going to remove, quote, the uninteresting variation, either with a rigid or an affine type of approach. If you ran the non-rigid registration to sort of do a highly squishable type of learning process before, then it's going to get a lot more complicated because it's going to pre-match so many of the features that you're caring about at that millimeter or 2 millimeter scale, and you're going to lose a lot of the subtlety. So I wouldn't do any of this after non-rigid registration, but the pose, I think, is generally a problem with the scan acquisition and scanner coordination.
So just to summarize, what do you recommend for this kind of investigation? Just to rigid transformation, or even, let's say, a fine linear registration?
I prefer rigid when it works, but affine tends to be much more robust. I think that conceptually the rigid is a better alignment, but when you are doing these kind of things and you have very different head sizes, you can get a sort of catastrophic failure of a rigid much more often, and you can get sort of an approximation on the local orientations and local angles with affine that you can deal with after the fact by sort of unwarping it. And I typically trade off the stability of the affine for the slight compromise on the local geometries and the relative positions.
Thanks a lot for your answer.
Do we have another question? If not, can I ask for some clarifications, especially for people who are starting now doing diffusion analysis? Can you explain a bit what is the infinite bandwidth B0 on the first method?
Let's see. With the fancy setup, it doesn't let me go very quickly on the slides. Get back here.
It's okay.
I'm getting there. So briefly, when you think about the bandwidth, that's how much time, how much of the signal frequency, the signal space you're using for each pixel. And so if you had infinite bandwidth, you'd basically be running one point at a time. You'd be just spending all your time getting one coefficient really well, and then do the next one, next one, next one. And the faster you go, then the less and less bandwidth you get. And so as you get lower bandwidth, the local inhomogeneities, the local problems become larger and larger. And so as you're down at 1 kilohertz, you can see the kinds of distortions that happen up here. And infinite bandwidth is basically, we went so fast that the distortions, that the local inhomogeneities were whatever size they were in kilohertz per pixel, divided by your bandwidth is 0. And so they were not distorted. I think we have a question.
Yes,
Probably time for one more.
I had a kind of clarification or maybe more detail about the transformation. So do you think that learning or having a network that is kind of translational and rotational invariant would be better? And why not use those type of model instead of manually registering? Because even if you do a rigid body, there's for example, if somebody have an hemisphere bigger than the other, they will not be aligned. So your goal is not to do the kind of nonlinear registration, but you assume that the affine is kind of